We generally like to think of science as, this reliable path forward, building knowledge step by step.
That's the ideal, certainly.
But what if that reliability? What if it's not quite as solid as we often assume? Today we're doing a deep dive into something called the reproducibility crisis.
Right, the basic idea being that researchers often struggle to get the same results when they try to repeat experiments that have already been published.
Exactly. It's unsettling. And there's this significant new study just out from Brazil that's adding some fresh fuel to this discussion.
Yeah, it's a preprint posted on bioxLarf in early April, so it hasn't gone through the whole formal peer review process yet, but it's from the Brazilian Reproducibility Initiative.
And it gives some pretty, compelling evidence about the scale of this issue, specifically looking at biomedical research there.
And what's really striking is the scale. We're talking 213 scientists, 56 different labs across Brazil involved.
Wow, that's huge.
Yeah.
Not just one team looking at one niche thing.
Precisely, it gives a much broader look. And their approach was interesting, too.
How so?
Well, instead of picking, say, famous studies or highly cited ones, they focused on very common, everyday methods used in biomedical labs in Brazil.
Like the routine stuff, things like the MTT assay, RT-PCR, what's the other one? Elevated plus maze.
Exactly, those kinds of techniques. The idea was to see how reproducible these fundamental building blocks actually are in practice.
That makes a lot of sense, getting at the foundations.
Yeah, it avoids certain biases and looks at potentially more widespread issues.
Okay, so our mission for this deep dive is to really unpack this whole reproducibility issue, using this big Brazilian study as our main lens.
I want to look at, is this new? What's the history? Is crisis even the right word?
And dig into the why. What's causing it? We'll definitely talk about the whole publish or perish thing.
Absolutely. So we'll cover the Brazilian findings in detail, glance back at the history, weigh the different arguments about whether it's a crisis, and explore those publication pressures and other factors.
All right, let's unpack it. So the Brazilian Reproducibility Initiative, what exactly did they do? How did they set it up?
Well, it was quite systematic. They started by reviewing a whole bunch of published life science papers from Brazil, just randomly sampled.
Right.
And from that, they identified the most commonly used experimental methods, especially ones using rodents or cell lines, because lots of labs would have that expertise.
Right, so they picked methods lots of people could potentially replicate.
Exactly. Then they selected 10 of these common methods. And the plan was, for each method, to aim for about 20 different experiments from published papers to be replicated.
20 experiments per method.
And crucially, each replication was attempted by 3 independent labs. That multi-lab approach is a real strength.
Sounds incredibly thorough.
Yeah.
But also complex, coordinating all those labs, especially, thinking about the last few years.
Oh, absolutely. The author specifically mentioned the COVID-19 pandemic threw a huge wrench in the works, logistically.
I can imagine.
And beyond that, there were disagreements sometimes between the different teams about, how strictly should we follow the original protocol? Are minor deviations okay?
The interpretation issues.
Right. One of the project leaders, Olavo Bor Amaral, he had this great quote. He said, it was like trying to turn dozens of garage bands, each with its own style, into a coordinated orchestra.
Huh. That perfectly captures the challenge, doesn't it? Getting everyone playing the same tune.
It really does. It highlights just how hard standardizing procedures across different environments can be.
Okay, so despite the hurdles they pushed through, what did they find? What was the big takeaway on reproducibility?
Well, the headline finding is pretty stark. Using their main criterion, which was that at least half of the replication attempts had to show statistically significant results pointing in the same direction as the original paper.
Okay.
Only 21% of the experiments met that bar.
21%? Seriously?
Yep. Just over 1 in 5.
Wow. So for 4 out of 5 findings they looked at using these common methods, they couldn't confirm the result in most of the labs trying to repeat it.
That's the basic picture, yes. It really makes you pause and think about the weight we give to single published studies.
No kidding. Was there anything else significant?
Yes, another really important point was about the effect size, basically, how strong the finding was. They found that the effect size reported in the original papers was, on average, 60% larger than what the replication labs observed.
60%. So the original studies weren't just hard to replicate, they were also potentially exaggerating the impact of the findings.
It certainly suggests an overestimation of effects in the published literature is quite common. That difference, that inflation, could itself explain why things are hard to replicate. The real effect might just be smaller and harder to detect consistently.
That makes sense. A smaller target is harder to hit reliably.
Exactly. And it's worth noting they actually used 5 different criteria to judge replicability. Depending on the definition, the success rate varied. I think it was between about 15% and 45%.
Okay, so how you define replicated really matters. It's not black and white.
Definitely not. It shows there's nuance here. And one final point on their methods. They had an independent committee validate the replication attempts. Out of 143 attempts, 97 were deemed valid.
So they had quality control on the replications themselves.
Yes, adding another layer of confidence in their overall findings.
Okay, so this Brazilian study paints a concerning picture right now. But is this whole reproducibility thing actually new, or have scientists always struggled with this? What's the history?
That's a really key question. And the answer is sort of yes and no. Large-scale systematic projects like this are fairly recent, say in the last decade or two.
Right, this level of organized checking.
But the underlying concern about whether scientific results are reliable and trustworthy, that goes way back. I mean, the history of science is unfortunately littered with examples of flawed or even fraudulent research. Think about things like the Tuskegee syphilis study or Vipe-Holm experiments.
Those were more about ethics, though, weren't they?
Right.
Horrifying ethics violations.
Absolutely. Primarily ethical failures, which led to crucial regulations like the Nuremberg Code and the Declaration of Helsinki. But underlying even those ethical debates is the assumption that the science itself should be sound and dependable. The need for trustworthy research isn't new, even if the focus shifts.
Okay, so maybe the way we measure reproducibility is new,
Yeah.
but the fundamental need for reliable science has always been there.
Exactly. The tools, the scale, the awareness might be different now, but the core challenge of ensuring scientific claims hold up to scrutiny is a long-standing one.
Which brings us to that term, reproducibility crisis. It sounds quite dramatic. Is it accurate? Is it really a crisis? Or could we see this differently?
That's the heart of the debate, really. On one side, you look at numbers like 21% from this huge study, and you look at previous studies in specific fields like cancer biology or ALS that found similarly low rates, sometimes near zero, sometimes up to maybe 40%. And it's hard not to feel a sense of, well, crisis. A crisis of confidence, at least. If results aren't reliable, it potentially misleads other scientists, policymakers, doctors, everyone who relies on that research.
Yeah, you think about wasted resources following up on false leads or basing treatments on shaky evidence. The potential consequences are serious.
Definitely. But then there's the other perspective. Is it a crisis, or is it science actually doing its job just more rigorously now?
How do you mean?
Maybe we're just better equipped and more willing to systematically check findings than we used to be. Science has always involved self-correction, refining ideas, overturning old findings. Perhaps this is just a more visible, more systematic version of that process. Growing pains, maybe, rather than a fundamental breakdown.
So a sign of maturity, almost. Science becoming more self-aware, more critical.
That's one way to frame it. It's also important, as we mentioned, that the Brazilian study, while large and method-focused, isn't alone. Its findings are concerning, but they broadly align with those earlier, smaller studies in specific disease areas. What makes the Brazilian one stand out is that it wasn't about one disease, it was about these common methods used across many areas of biomedicine in that country. That wide lens makes the findings perhaps more broadly indicative.
That cross-cutting approach does seem particularly powerful. Okay, let's dig a bit deeper into the specifics of what the Brazilian study found. Were there particular patterns in why things didn't replicate?
Yes, and this is really interesting. They found that the differences in effect size, how big the effect was, were generally larger when you compared the original study to the replications than when you compared the different replication attempts to each other.
Huh. Okay, what does that suggest?
It strongly suggests there's something specific going on with the originally published articles. Things like publication bias, the tendency for journals and researchers to favor publishing positive, statistically significant results over null or negative ones.
Right, the stuff that makes a better story.
Exactly. Or selective reporting, maybe researchers only report the experiments that work best. Or maybe the original studies just didn't have enough statistical power, enough data points to get a reliable estimate of the effect in the first place. And of course, the whole incentive structure rewarding splashy findings.
That links back to the publish or perish idea we mentioned. Pressure leading to maybe. overly optimistic reporting.
It certainly points in that direction, but, and this is also really important, they did find significant variation among the different replication labs, too.
Ah, so it wasn't just about the original papers being different.
No. It tells us that even when multiple labs try to follow the same protocol carefully, other factors come into play. Tiny differences in lab conditions, reagents, equipment, even how individual researchers interpret ambiguous steps in a protocol.
The garage band versus orchestra problem again.
Precisely. Replicating complex biology perfectly across different settings is just inherently challenging. So there are issues potentially originating from the published record and issues inherent in the practical process of replication itself.
It's coming from both ends, it sounds like. Did they mention anything else about the data itself, like how variable it was?
Yes, they looked at the coefficient of variation, which is basically a measure of data spread relative to the average. They found that in most cases, especially for those MTT and PCR experiments, the CV was lower in the original studies compared to the replications.
Lower variability in the originals. What might that mean?
Well, it could suggest that the original published results might sometimes present a cleaner, less noisy picture than what typically happens when you repeat the experiment. Again, potentially tied to selecting the best data or conditions for publication.
Making things look a bit tidier than reality.
It's a possible interpretation. One slightly reassuring note, though. They found that sign errors, where a replication got a significant result but in the opposite direction to the original, were actually quite rare.
Okay, so it's more about the size of the effect or whether it shows up at all, rather than things being completely backwards.
Generally, yes. That seems to be the more common pattern based on their findings.
Still, it leaves us with this significant replication problem. And we keep circling back to publish or perish. How did that culture become so dominant in academia?
It really took hold, historians argue, after World War II with the massive expansion of universities and government funding for research. Before that, research was important, but perhaps not the sole focus for many academics.
Right, there was maybe more emphasis on teaching or other roles.
Exactly. But as universities grew and competed, research prestige, measured primarily by publications in peer-reviewed journals, became codified as the key metric for hiring, for getting tenure, for promotion.
So your career literally depended on racking up publications.
Pretty much. It led to this counting mindset. How many papers do you have? What are the impact factors of the journals? It became paramount.
And this pressure affects funding too, right?
Hugely. Grant applications are heavily judged on your publication record. So you need publications to get grants, but you need grants to do the research to get publications. It's a cycle of intense pressure.
And that pressure, it can lead to some questionable practices, presumably. Not necessarily fraud, but cutting corners.
Unfortunately, yes. It can incentivize things like slicing research findings into the smallest possible, least publishable units, just to get more papers out.
Salami slicing.
Right, or repackaging the same basic data for multiple publications. Or adding people as co-authors who didn't really contribute substantially just to boost everyone's numbers. The focus can shift from the quality of the science to just the quantity of output.
And that inevitably impacts reliability, you think?
It certainly can, and most directly related to reproducibility, this pressure creates a powerful bias towards wanting positive, statistically significant results, because those are often easier to publish, especially in high-impact journals.
So researchers might, consciously or not, select data, tweak analyses, or interpret results in a way that favors finding significance.
That's the concern. The system might inadvertently reward practices that aren't aligned with producing the most robust, reliable findings. It's a structural issue.
It really sounds like a systemic problem. Beyond publish or perish, though, what other factors contribute to experiments being hard to replicate? You mentioned protocol issues earlier.
Yes, that's a huge practical barrier, especially in lab-based biology. The methods sections in papers are often just incomplete. They lack crucial details.
Like the exact temperature or the specific brand of a chemical.
Exactly. Or subtle details about how cells were handled, or the specific settings on a machine. It makes it incredibly difficult for another lab to replicate precisely what was done. The Brazilian teams often had to contact original authors for clarifications, and even then, information was sometimes missing.
So you can't even be sure you're doing the same experiment.
Pretty much. And there's also a lack of standardized terminology. How do we describe controls? Randomization, blinding. Different labs might use terms differently or just not report these critical design elements clearly.
Just basic reporting standards aren't always there.
Right. And then there's the role of the journals and the publishing industry itself.
How do they fit in?
Well, the rise of large commercial publishers means journals are often run as businesses. While they facilitate dissemination, there can be inherent conflicts. The traditional subscription model limits access, which hinders replication.
If you can't read the paper, you can't replicate it.
Exactly. And while open access aims to fix that, the high article processing charges, or APCs, needed to publish open access can be a barrier for researchers without big grants, especially in developing countries. It creates inequities.
So access to both read and publish can be an issue.
Yes, and then there's peer review. It's crucial, but it's not perfect. There can be biases against certain institutions, maybe against novel ideas that challenge orthodoxy, or even against negative or null findings.
Reviewers might prefer results that confirm existing theories, perhaps.
That can happen, and sometimes researchers, especially when talking to the media, might, let's say, sensationalize their findings a bit, extrapolate beyond the data. That can create unrealistic expectations, too.
Setting things up for a perceived failure later if replications are more modest.
Precisely. It's a whole ecosystem of interacting factors, the incentives, the reporting standards, the publication system, the practical challenges of lab work.
It really is a complex web. So bringing this all together, the Brazilian Reproducibility Initiative feels like a really important piece of evidence, doesn't it? Giving us that large-scale, method-focused look.
Absolutely. It provides a very concrete, data-driven snapshot of the challenges in one major scientific context, and it reinforces the idea that tackling this requires a multi-pronged strategy. It's not just one thing.
You can't just tweak one part of the system.
No, you need to think about improving methodological reporting, maybe changing academic incentives, reforming aspects of publication, promoting more rigorous training. It's a big task.
It definitely feels like a critical moment for the scientific community to reflect and act. So, thinking about everything we've discussed, what do you see as the most crucial steps? If you had to point to one or two things that institutions, funders, scientists themselves need to prioritize to really strengthen the reliability of research going forward, that's the big question to leave our listeners with.
It is the question, isn't it? And there's no single easy answer. But thinking about where to start is vital for science's future.
Absolutely. Well, thank you for diving deep into this complex topic with us today. We've really covered a lot: the striking findings from the Brazilian study, the history behind these concerns, the whole crisis debate, the huge impact of publish or perish, and this whole range of other contributing factors.
From missing method details to journal economics.
Exactly. Hopefully this gives you, our listeners, a clearer picture of the challenges and the ongoing conversation. If you want to dig deeper, do look up the Brazilian Reproducibility Initiative's work. The preprint is on Bioxer, and there was coverage in journals like Nature.
Understanding the problem is definitely the first step towards finding solutions.
Indeed. Join us next time on The Deep Dive, where we'll explore another fascinating topic shaping our understanding of the world.